Hello, and thank you for attending this talk from Quanterix on the value of ultrasensitive Simoa technology for non-invasive biomarker research. To begin with, I would like to say a few words about Quanterix's goal as a company, and that is to facilitate predictive and precision medicine by enabling researchers to measure very low levels of molecules that can serve as biomarkers indicative of different pathologies before symptoms even occur, and also to enable that to be done in easily accessible sample types such as blood, saliva, urine, and others. And how we do that is very interesting. We also use antibody pairs like ELISA does, but we get to femtogram per milliliter levels of sensitivity. And we have found two creative solutions for that. One of them is by building the ELISA immunosandwich on a bead. These beads are then isolated across femto-size wells, and then the volume inside this well is so small that the fluorescent product generated by the enzyme will be very concentrated, enabling us to see a single molecule. That's why we call this oftentimes as digital ELISA. On the other hand, many times multiplexing is of great value, and that's why we have an alternative technology in which we get the same levels of sensitivity by using capture antibody spots printed and deposited at the bottom of the well. We then build a monocomplex on the surface of the spots, and we take advantage of the circular pattern of the spots, which then is favored by the fluid dynamics to ensure a higher capture efficiency. And we also use chemiluminescence that has a very low intrinsic background and allows us to play with the exposure times to capture as much as possible from low analyte samples and also from high analyte samples, enabling us to have a really good dynamic range and multiplex at the same time. I cannot talk about Simoa without mentioning neurology. And the reason Simoa has revolutionized the neurology field is because it is now possible to measure these brain proteins directly in blood. And I have two case studies to illustrate that. The first one on multiple sclerosis, in which many researchers have shown that this one of the biomarkers we have called NFL will correlate with disease activity and severity, as was observed in comparing that with MRI. It correlates, as you see on the first graph, with new lesions and total lesion load on the MRI images. And that's why it can serve to monitor treatment response in a very easily accessible sample type. On the other hand, we also have for the first time an Alzheimer blood test that it's based on the biomarker phospho-tau 181. This test can distinguish dementia coming from Alzheimer's from other kinds of dementias. It can also identify Alzheimer's specific pathology that's usually done with PET scans, very expensive. And it is even prognostic in individuals with mild cognitive impairment before Alzheimer develops. All the other neurological conditions you can think of will have applied Simoa, including COVID-19 and its neurological manifestations. And talking about COVID-19, there was a lot of contributions of the technology to understand SARS-CoV-2, starting with the type 1 interferon response and observing how quantifying interferon alpha levels, even trace levels of it in blood, was very helpful in distinguishing the different manifestations of disease and understanding that severe and critical patients have an impairment in type 1 interferon response. Of course, you can imagine that cytokine profiling with ultrasensitivity is also of great value, as it can serve as prognostic cytokines of a risk of developing a severe disease, for example, as hyperinflammation is very well known in COVID. And finally, we have a very new application from two new tests that we developed, one for ultrasensitive quantification of the antigen protein of SARS-CoV-2, and another serology test that quantifies IgG binding to SARS-CoV-2. You can see here on these figures that we actually are even able to quantify that in dried blood spots and saliva. We also have shown to be possible in this publication, even in asymptomatic individuals. Seroconversion we can also observe very early, and it also correlates with these patients who recover. So I hope I could provide you some examples of how Simoa can be used. I am available for questions, and thank you very much for attending this talk.